Good morning, students. I am Ms. Republica Borgohain, Assistant Professor, Assam Downtown University, Guwahati, Department of Pharmaceutical Science. So today we will be going to discuss about the Fischer's lock and key model of enzymes. So we know that Fischer's lock and key model, it's a template model and it is proposed by Emil Fischer in the year 1898. The union between the substrate and the enzyme takes place at the active site, more or less in a manner in which a key fits a lock and results in the formation of an enzyme substrate complex. So enzyme, it reacts with the substrate and forms the enzyme substrate complex. So this is the reaction which has been shown below. In fact, the enzyme substrate union depends on a reciprocal fit between the molecular structure of the enzyme and the substrate. And as the two molecules, that of the substrate and the enzyme, are involved, this hypothesis is also known as the concept of intermolecular fit. The enzyme substrate complex is highly unstable and almost immediately this complex decomposes to produce the end products of the reaction and to regenerate the free enzyme. The enzyme substrate union results in the release of energy. It is this energy which is in fact raises the energy level of the substrate molecule, thus inducing the activated state. And in this activated state, certain bonds of the substrate molecule becomes more successive cleavage. Now the Koshland's induced fit model. It is unfortunate feature of Fischer's model and is the rigidity of the active site. So Koshland presumed that the enzyme molecule doesn't retain its original shape and structure, but the contact of the substrate induces some configurational or geometrical changes in the active site of the enzyme molecule occurs. Consequently, the enzyme molecule is made to fit the configuration and active centers of the substrate. At the same time, other amino acid residues may become buried in the interior of the molecule. As to the sequence of events during the conformational changes, three possibilities exist. That is, the enzyme may first undergo a conformational change, then bind substrate. Secondly, an alternative pathway is that the substrate may first be bound and then a conformational change may occur. Thirdly, both the process may occur simultaneously with further isomerization to the final conformation. Koshland's model has now gained much experimental support, and these changes during substrate binding and catalysis have been demonstrated for various enzymes, that is phosphoglucomutase, creatine kinase, and carboxypeptidase. So here how the substrate and the active site reacts in this model. Now about the activation energy. All the chemical reactions in a biological system have an energy barrier which prevents reactions from proceeding in an uncontrolled and spontaneous manner. The input of energy required to break this energy barrier or to start a reaction is called the activation energy. Enzymes lower the activation energy of a reaction, that is, the required amount of energy needed for a reaction to occur. They do this by binding to substrate and holding it in a way that allows the reaction to happen more efficiently. So this is the chart for the activation energy. We also know that it is important to remember that enzymes do not change the reaction's G part. In other words, they do not change whether a reaction is exergonic, that is spontaneous, or endogenic. This is because they do not change the reactants or products' free energy. They only reduce the activation energy required to reach the transition state. So this was the I conclude this enzyme chapter with the activation energy which is required mostly for the lock and key model and the Koshland's induced fit model. And the most important part is the role of the activation energy. So here I conclude with my slides, my topic. So in my next video I will be discussing about what is coenzymes. Thank you.